So today is October 9th, 2020, and the topic of today's lesson is translation. Yesterday we talked about transcription, so we're going to the next step in this process of protein synthesis today. Today is Unit 5, Day 4. Our objective is to explain the steps involved in the process of translation, in which mRNA codes for a chain of amino acids. So now we're going from mRNA to amino acids. Our essential question today is, how does a sequence of nucleotides code for a sequence of amino acids? I put the word sequence in bold and italicized the word sequence as well because it is so important when we think about structure determining function. In this case, sequence is structure. So if I change the sequence, then I change the function. All right, sequence is so important in this lesson. Keep that in mind. So at this point, you should have your notes out. Hopefully you've written down the title of today's lesson. Hopefully you've also written down October 9th, 2020 in Unit 5, Day 4. I will give you 5 more seconds to do that. All right, moving on. So yesterday we talked about the process of going from DNA to mRNA, and that process is called transcription. Today we're talking about going from mRNA to amino acids, which is a process called translation. Okay, this is all a part of the most central dogma of biology, the most important concept of the study of life, which is that DNA codes for the proteins that we make, that our cells make. Meaning DNA is the instruction manual to make all of the different proteins that carry out most of the functions needed for life. So this is an unbelievably important concept. Today we're talking about the second part of it, which is translation. Okay, so remember, we should be taking notes on the things that are highlighted, which means I need to highlight one more thing. So the goal of translation is to link amino acids together to start making a protein. Actually, I don't like that definition. I'm reading it. Say something different. To read the mRNA sequence and begin. All right, yeah, I just added that. That's better. Sorry about that. So the goal is to link, oh, I messed up there. So the goal is to read the mRNA sequence and begin linking amino acids together to make a protein. This is all messed up. Oh my gosh.
What does that say? I'm trying to read it, but there's a whole bunch of words in the way.
Yeah, give me 1 second. Yeah, that should work. Ooh, okay, I had to get that formatting out of the way. If you haven't noticed, I'm a bit of a perfectionist when it comes to formatting. Now, it's easy to remember that transcription happens first because transcription and translation are ordered in alphabetical order. So transcription is alphabetically before translation because C comes before L. Transcription, then translation. So translation is the second step. It comes after transcription. Translation occurs in ribosomes.
For some reason, when I look at that picture, I see a monster eating those. And that was his teeth.
Yeah, it does look like teeth. Okay. So I want you to write down the steps, focusing on the words that are highlighted. I'm going to give you a lot of time to do this. Let's say 3 minutes. That might be way too much, but I'm going to give you 3 minutes to write this down without talking about it. Okay, halfway, minute and a half left.
When the timer's up, you can explain like what all this means.
Yes, ma'am.
Okay.
Five seconds. 4, 3, 2, 1. Okay. So the first step of translation. If you remember from transcription, we end up transcribing an RNA strand, an mRNA strand. That mRNA strand leaves the nucleus and it just goes out into the cell's cytoplasm. So the first step of translation is that the ribosome will attach to that strand. Once the ribosome attaches to that strand, it reads the strand, and it reads it in a special way. It doesn't just read it letter by letter, it reads in groups of three letters, aka 3 bases. And we call these groups of 3 bases codons because they code for something specific. Either they code for an amino acid. We have 20 amino acids. So either they code for one of those 20 amino acids, or they code for a stop message, which I'll explain later. But the stop message signals the end of transcription. So the ribosome is reading the mRNA strand in groups of 3. And as it reads it, a tRNA molecule is going to bring a specific amino acid to the ribosome. The way that tRNA molecule is able to do that is that each tRNA has a specific anticodon, which we'll explain on the next slide. It has an anticodon that corresponds perfectly to the codon. So it will bring, that tRNA will bring the amino acid, the correct amino acid, to the ribosome. And then our last step is that those ribosomes are linked together with peptide bonds, creating a polypeptide chain. Poly is a prefix that means many. So a polypeptide chain is a chain of many peptide bonds. And eventually that polypeptide chain is going to fold together in some very intricate ways to make a protein. So these are the 4 steps of translation. Let's look at this image. It might help explain it. So we've got our ribosome here, our blue ribosome. We've got our strand of mRNA, which you can see. You can see how the bases are grouped together in groups of 3. We call that group of 3 a codon. So here's one codon. Here's another codon. Here's another codon. What you should notice is that there are these gold structures that have this weird shape. That's your tRNA. At the bottom of each tRNA is an anticodon, which corresponds perfectly with the codon, right? So I've got, in this case, I've got a codon ACG. So the anticodon of that is U, because A would then be paired with U, G, because C is paired with G, C, because G is paired with C. So ACG is paired with UGC. ACG is your codon, UGC is your anticodon. And so this is how the tRNA knows, okay, well, it's my turn to come now into the ribosome because I have the correct anticodon. So the anticodon here would be AGU. We need a tRNA with an anticodon of AGU, and it's going to bring another amino acid. And they continue to get linked here, forming a polypeptide chain. So here are the steps again. Step 1, the ribosome attaches to the mRNA. That's what it says down there at the bottom. It's blocked out right now, but the ribosome attaches to the mRNA. Step 2, the ribosomes read codons. So they read these bases in groups of 3. Step 3, tRNA, which literally looks like a lowercase letter t, but it stands for transfer RNA. The tRNA with the correct anticodon will bring its amino acid to the ribosome and prepare for it to be linked. And that's the last step. You end up with a chain of polypeptides that starts to form.
Were we supposed to draw this?
No. Are there any questions about this process? It's okay to have questions. This can be confusing stuff.
Why is the tRNA really shaped?
It has to do with the fact that there are different properties, just like our cell membrane, for example, has a hydrophilic portion that likes water and a hydrophobic portion that does not like water. So because of that, the hydrophobic portion exists on the inside so that it does not have to interact with water. So we can see how the properties affect the shape. The properties of the membrane affect the shape. Same thing with this tRNA molecule. The properties These are actually, the tRNA, of course, is made up of these same nucleotide bases, UACG. So these tRNA molecules have properties that affect that determine what their shape will be. It's more so chemistry than it is biology. Good question though. Anything else? Any other questions?
What is that thing called in the background? That the green thing.
This big thing and this big
Mhm.
colorful thing. That's the ribosome.
Oh, I thought the ribosomes were pointing to something else.
Well, we know that the function of ribosomes is to make proteins, right? So that's what's happening here. This ribosome is making a protein by linking these amino acids together. Also a good question. Anything else?
Oh,
go ahead.
Can I ask another one? I see that tRNA is like on top of the anticodons. What is that like? What does that do?
Okay, so
like, are they connected?
So this is going to be a good image to show for that question and for the question that Alicia asked. Okay, so the anticodon is literally just a group of nucleotides that faces outwards. So we can see there are three different ways of looking at this structure. This is a 2D model of a tRNA. There's a 3D, it shows some of the protein, some of the folding of the tRNA, and here would be a space fill model. We can see that these are A's and C's and G's and U's. So this is the exact same tRNA, just shown in different ways. Alicia's question was, why is it shaped like that? It's shaped like that because these nucleotides in the straighter sections of the tRNA are bonded together. In the loops, they are not bonded together with anything. And then down here at the bottom, we've got an anticodon that faces outwards. We've got a single three nucleotides that face outwards here rather than facing inwards towards the rest of the tRNA. And so it's not that the tRNA is on top of the codon, it's just that the codon is a part of the tRNA. It's attached to the tRNA. It is the tRNA. It's the bottom of the tRNA. So here, this is a simplified version of what we just saw. But these anticodons, like in this case, we've got an anticodon UCA, that's attached, that is a part of this tRNA. And it helps the tRNA to read the codon. So let me put it like this. Hold on. Come on. This is difficult stuff, so I'm glad you guys are asking questions. I'm going to draw something very, very basic here. Just imagine that this is a tRNA. And I've got a codon UCG. Then I've got another tRNA, and this has the codon AUA. And I've got another codon, tRNA, and it has the anticodon GU. A. So these are all anticodons. These are tRNA molecules. Now let's say that I have let's say that I have a codon of mRNA that has, of course, 3 bases. And that codon is UAU. Which one of my tRNAs corresponds to that codon? Which one of my tRNA over here has the right anticodon for this codon?
Isn't it the middle one?
The one in the middle. Oops, I'm sorry. Yeah, the one in the middle. Even though I have all these different options, I've got 3 different options. The one in the middle corresponds to this one because its anticodon is AUA. So A matches with U matches with A matches with U. So this would be the right tRNA for this codon. And each of these codons has a slightly different give me a second. Each of these codons has a slightly different amino acid. So when this tRNA is chosen, when it knows that, okay, my anticodon matches with this codon, it's going to bring with it this green amino acid here. And so that's how mRNA codes for a sequence of amino acids. The mRNA sequence here, UAU, matches perfectly with the tRNA that carries the right amino acid. That's the complexity, but also the specificity of this process. Right? So if this amino acid, if this tRNA had tried to bring its amino acid, it wouldn't have worked because UCG does not match with UAU. Does this make sense?
Yeah. Now that you explained it more in depth, it makes sense. Sorry you had to explain it more in depth.
No, that's okay. And are there any more questions? Because this is a little difficult. So each tRNA has its own, it's going to have its own amino acid, and it's going to bring that amino acid with it when its anticodon matches up with the codon. That's the takeaway here. Now, you won't really be tested to that level. It's good to know what's going on in this process, but really you just need to know in general what happens in translation. Translation is the process of reading an mRNA strand, and eventually you start linking amino acids together. That's basically the extent of what you need to know here. Okay, so we're going from the language of nucleotides, AUGC, that's the language of nucleotides, nitrogen bases, to a language of amino acids. So we're translating languages here. In the past, we did transcription. We transcribed from DNA, which was still nitrogen bases, to mRNA, which is still nitrogen bases. All right, so same language, we just wrote it slightly differently. Here, we're writing a different language. So this is Hebrew for you are the most amazing students ever. Miss Baxley speaks Hebrew. So, all right. So we've watched this video before. Perhaps it'll make more sense now. Actually, no, I don't want to spend time doing that right now. All right. So, go ahead and write this down. In nature, there are 20 amino acids.
I have a question. What about you know how like they have amino acid drinks or something? Does that give you more amino acids or like what
I've
is
never
it?
heard of an amino acid drink.
No, not an amino acid drink. There was this lady who made moringa lemonade. Basically, it's like lemonade from a plant, and she said it had amino acids in it. Like, does she mean the amino acids we're talking about, or something totally different?
Well, again, amino acids are just the monomers for proteins. So anything that has proteins will eventually be broken down inside of your body, and it gets broken down into amino acids. So I guess if it's a protein shake, maybe that's what she meant.
I don't know. Maybe.
Okay. So we've got 20 amino acids, and every codon codes for a specific amino acid, or it codes for a stop message. So 2 codons code for a stop message, which means that translation needs to end here. This is the end of our polypeptide chain. Those 2 codons are UAA and UAG. Those are your stop messages. Now, there's an order here, and we're going to talk about how you read this chart to figure out which amino acid you're dealing with. So there's an order. First base is on the left side, second base is on top, and the third base you read from the right. So this is why this sequence matters so much. It cannot be overstated. Even a small change in the sequence can lead to a totally different amino acid, a totally different message. And each amino acid is slightly different in the properties that it has. So if I make the mistake, I'm supposed to have a hydrophilic amino acid, or an amino acid that likes water, but then I end up making a mistake. I have some type of mutation, and I code for a hydrophobic amino acid, one that does not like water. That can be extremely problematic. It can mess up the way that the protein folds, and it can mess up the function of the protein. So when you change the sequence, you change the function. Okay. Any questions about this so far? Now, one thing to keep in mind, I'm going to push you all a little bit here. We have 20 amino acids, but we have way more potential combinations than just 20. So in theory, I could have more than 20 amino acids. If every single codon coded for a different amino acid, then I could have 64 amino acids instead of 20. But we can see that some codons code for the exact same amino acid. For example, UUC and UUU both code for phenylene. We can see that CUU, CUC, and CUA and CUG all code for leucine. They all code for the exact same amino acid. What would be the reason for that? Why do you guys think our cells have adapted to do that? Where we can get the same amino acid from different codons. Any ideas about why that is?
Is it so the process moves faster?
Okay, that's a big guess. It's about the speed. Okay, Gabby says genetic variation. Think about what I just said, though, in terms of if I make a small mistake, it could have profound impacts on the function of the protein. So if I'm supposed to have, so let's just imagine that CUU and CUC, these two were actually different. CUU gives me leucine, but CUC gave me something different. That would mean that if I just change one letter, it could totally throw off the function of the protein. So the benefit of having multiple codons code for the exact same amino acid is that you're less mistake prone, right? Even if you do make a mistake, it's not catastrophic, right? So even if my cell makes the mistake of putting a U where there's supposed to be a C, or putting an A where there's supposed to be a G, it doesn't totally ruin the protein. I end up with the same amino acid. So this is kind of an ingenious evolutionary development of our cells. The same amino acid can be resulted from different codons. You guys will better understand that based on what we talk about in the coming lesson. So we've got two codon charts. Remember, a codon is a group of 3 letters, a group of 3 bases. So these codon charts help you determine what those 3 bases result in, which amino acid those 3 bases will give you. Either one can be used. It's just a matter of preference. When I was in high school, I was only taught to use the chart on the left, but in college, I learned the chart on the right, and I prefer the chart on the right now. So it's just a matter of preference to you. Let's talk about the one on the left first. So this is not highlighted, but you may want to write it down. Don't write it down now, just listen to what I'm saying, and then I'll give you time when I'm done talking to write it down. So first, you want to identify your codon. And just as an example, we're going to use GAC here. Second, you want to identify the first base of the codon. So whichever letter comes first, whichever base comes first. In this case, that of course is G. I'm using the color red for G here. Then you want to find the horizontal row of that first base. So I want to find the row over here. We see that it says first base over here. So I want to find the row that corresponds with my first letter. In this case, my first letter is G. So I'm down there in that bottom row. That means that I can really stop paying attention to everything up here. None of this is going to be an option for me. I can only have one of these amino acids in this bottom row. Now I need to look at my second base of the codon. So in our example, this second base is A. I've got A right here. All right, so I need to look up in my second base and find the right column, so my vertical column for the second base. It's not U, it's not C, I've got A. So I'm looking at this column right here. It's the color green for A. And then lastly, the overlap between the row and the column is where my amino acid is located. So now I'm not even looking over here anymore. It can't be valine, it can't be alanine, it can't be glycerine. I'm only looking in this section right here. My third base is C, that's the color blue for C. Now I want to look on the right side, and I can see the letter C here. So that means that I'm going to use an arrow, and that tells me that ASP is my amino acid. So we started off with a codon, GAC, and that codon gives us the amino acid, aspartic acid, which is ASP abbreviated. So now is the time to write down these directions. Let's take, you don't need to write down the examples, but let's just take 2 minutes to write this down, if you think you want to use this chart.
Somebody whistling?
Sorry.
Oh, my bad. I'm sorry. Do you wear glasses? You don't wear glasses, do you?
Yeah, I do.
Okay, I wasn't sure if you wore glasses or not because I felt like you wore glasses, but then you're not wearing glasses now. I'm confused. Does he wear glasses?
Yes, I decided to take the morning off from my bifocals.
My brother, he wears glasses too. Well, he wears reading glasses.
That's what they say, the people who read the most, your eyes go first.
Then how come I don't have reading glasses? I read all the time.
I guess you're just genetically blessed.
I don't know. I went to the doctors and they said I had better than 20/20 vision.
Superhuman. Okay, that was 2 minutes. So this is just one of the codon charts. The other one is also available to you. It's just a matter of preference which one you use. We're going to stick with that exact same example, so we should end up with the exact same amino acid. We should end up with ASP because we're using the same amino acid. I'm sorry, the same codon. So we're sticking with GAC. Here's how you use this one. Looks complicated. It's not nearly as complicated as you think. Again, you need to identify what your first base is. In this case, it's G. So starting from the middle, start from the middle. Find the bases section of the pie. So find G's section of the pie. Now, once I find it, I can ignore all other sections. So there's G's section. I can ignore those other sections. They don't matter anymore. So my options are now glycine, glutamic acid, aspartic acid, alanine, and valine. Those are my only options. If I've started off with the first base correctly, I've limited my options. Now I need to identify the second base, which is in this case A. I'm going to find the section of the pie for that second base now. So I'm moving outwards. Start in the middle, I'm moving outwards. Here's the second section of the pie. All right, so I've now limited my options again. Now I'm only left with glutamic acid and aspartic acid. And then my third letter is C. So now I need to find the section of the pie for C, and there I am. Aspartic acid. It's the same answer we got as before. So that's how you know you did it correctly. You can find it on both charts. It's good.
I'm confused on what we just did.
Thank you.
What were we supposed to be identifying?
So this whole thing is about identifying the amino acid. Our amino acids are on the outside here. So we're going to start in the middle and move outwards. What's my first letter here, Ayana?
G, I guess.
G. Yeah, G is my first letter. It's my first base. So which section, top right, top left, or bottom right, am I looking at?
Top left.
Top left, right. There's my G right there. So I'm only looking in the top left. What's my second letter, Ayana?
A.
A. So does that mean I'm looking here?
No.
No, because that's G. Should I be looking over here?
No.
Nope, that's C. I should be looking here where there's an A. All right. So the other sections don't matter anymore. I'm just looking at A. Now, what's my third letter, Ayana?
C.
C. Now, do you see our four letters here again? U, C, A, G. Do you see them?
Mm-hmm.
Which one, where's the C? Is it in this section with glutamic acid, or is it in this section with aspartic acid?
It's with aspartic acid.
Exactly. So that's your amino acid. So GAC codes for aspartic acid. Good job.
Thanks for explaining that.
You're welcome. Here, let me push you a little bit further, and this doesn't just have to be for Ayana. Anyone can answer this. What else codes for aspartic acid? What's the other codon that codes for aspartic acid? I feel like someone's trying to say it. Okay. So look, remember, a codon is a group of 3 letters. Here's aspartic acid right here. I can work backwards from aspartic acid. I can see that in order for me to have aspartic acid, I need the first letter G. I need that G. I need the second letter A. My third letter can be either C or U. Either C or U can be the third letter. So GAC gives me aspartic acid, but the codon GAU also gives me aspartic acid.
I'm so dumb.
Do you see that now? Okay. Well, we just need more practice. All right, so I'll do this first one using this chart. Wait, what happened? It should be, okay, sorry. Should say I do. What happened? Nope. So my mRNA codon is AGU. So my first letter, I'm looking over here now, my first letter is A. So that means I'm already in this section. My second letter is G. So I'm looking at the top now. My second letter is G. So now I'm looking in this column here. My third letter is U. So I'm looking right there. That tells me serine. Serine is my amino acid. Let's try another one. GAC. G is my first letter, so I'm looking in this column, this row at the bottom. A is my second letter, so I'm looking in this section right here. And then C is my third letter, so I'm looking right here at aspartate or aspartic acid. That's the same ASP. Okay. Can anybody tell me what CAG codes for? And walk me through how you do it. Someone volunteer for us. Someone, anyone. Come one, come all. CAG, folks. Come on, someone give me a try.
Doesn't it
I'll walk
code
you through it.
I know it starts with a G.
What's the second letter? I mean, like, spell the word for me.
I don't know how to spell it. It's just in my head. It starts with a G.
Okay. How do you know? Can you see the screen?
Mm-hmm.
Okay, so what's the first letter of this codon?
C.
Okay. Second letter is?
A.
So what are my two options in that case? I'm looking right here.
Histidine and glutamine.
Excellent. And then my third letter of the codon is what?
G.
Okay, great. So that means that it has to be which of those two?
Oh, glutamine.
Exactly. Good. So we're looking right here. It's glutamine. Good job.
I think I just gained some knowledge.
Sounds like it. Okay, thank you, Ayana. Can someone else volunteer for us? If you need help, I'll walk you through. We're looking for AUC here. Someone volunteer for us, please. AUC. Unless someone is in the chat telling me what it is. Oh, what the heck? Why don't you guys take yourselves off of mute, please? Okay, thank you, Olivia. That is correct. AUC is isoleucine. Let's try another chart. Who can do AGU for us using this chart?
Serine, I think.
Good. This is serine. What about GAC? Someone tell me what GAC is. We actually did that already.
Aspartic acid.
Yep, that's aspartic acid or aspartate. Same thing. Okay, let's put it all together here. This is the last thing we're going to do today. So I first need you to transcribe from DNA to mRNA, and then you're going to tell me the amino acid. That should say amino acid, not codon. So you're going to transcribe from DNA to mRNA first, and then you're going to translate from mRNA to amino acid second. So transcribe T, what does it become?
A.
A?
U-G.
AUG. So there's my codon right there, AUG. Let's keep going though. What does G transcribe to?
C.
C,
C.
C,
G.
G. Then what?
A-U-C.
Then what?
U-A-A.
Excellent. All right. So now we've got our codons. AUG, CCG, AUC, and UAA. Let's start with this first one. AUG, what does that give us?
Methylene.
Methionine, yeah. MET. Now, fun fact, almost all of these polypeptide chains will begin with AUG. They'll begin with methionine. Methionine is considered the start amino acid. AUG is considered the start codon. That's not universal, but that's what often will happen. Okay, CCG.
Pro.
C, C, G. That gives us proline. Good. What's next? AUC.
L-L-E.
Good. ILE. Sorry, it's an I. It's not. It looks like an L, but it's an I. And then our last one, UAA.
Stop.
Yes, good. That gives us a stop. UAA gives us stop. All right. So obviously we're using abbreviations for these amino acids. It's fine if you use the abbreviation. Sometimes you'll see them as just one letter. We won't really introduce that here. You don't need to memorize the amino acids. You don't need to memorize the chart. You just need to know how to read it. Okay, there is an activity. There really are two activities on Canvas. Of course, there's an exit ticket. You should probably start with that. There's an activity called translation practice. It has two questions that basically walk you through this activity where you have to transcribe, then translate. And then it also is associated with a Gizmo. So if you still haven't made a Gizmo account, you're going to need to do that. At this point, because translation is a difficult thing to do, these codon charts are not really easy. I will say that if you need some additional help, please do not hesitate to make an appointment with me to meet one-on-one so we can walk through these. Some of you are being a bit shy and don't want to ask questions during class, or you don't want to volunteer so that I can help you a little bit. And that's okay. I'm a shy. I was at one point too. But we got to figure out how we can provide some assistance to you. We don't want to let this go on too long without you knowing where we are in the class. So make an appointment with me one-on-one. If you feel like you need some help, I'd be happy to explain these concepts to you in a more personalized way. All right, but I will leave you all to it. We've only got 6 minutes left in class. There are now two gizmos that you should have taken the assessment for. There's the building DNA gizmo, and there's the RNA and protein synthesis gizmo, which is the one that today's lesson is associated with. All right, so I'll leave you guys to it. We've got 5 more minutes. Okay, it's 11:35, so I will let you guys go, and I hope you enjoy your weekend. Please let me know if you need any assistance over the weekend. We do have midterms next week, so I want to make sure you all are in a good place to succeed. I will be available to answer questions, and I'll talk to you soon.
Oh wait, I have a question. I don't think
Yeah.
I'll be setting a meeting